Hello, my name is Miganush Stepanians. I'm the president, CEO, and founder of PROMETRIKA, and a drug development focused biostatistician with 30 plus years of experience. PROMETRIKA is a full service clinical research organization located in Cambridge, Massachusetts. We have been collaborating with our sponsor companies in the development of hundreds of drugs and biologics, including many treatments for rare and ultra-rare conditions. So this presentation is purely from a statistician's perspective. Throughout the years, I have collaborated with multiple biotechnology companies in more than 30 rare and ultra-rare indications, and these collaborations have afforded me the opportunity to interact with the FDA and European regulatory bodies on rare disease trial design and analysis. So what are the challenges of rare disease trial design and analysis? Well, first and foremost, we have very small sample sizes, especially in ultra-rare, but also, even maybe more importantly, the heterogeneity of manifestations of disease that are present in these rare and ultra-rare indications. Finally, challenges that can be summarized as a lack of precedent, a lack of regulatory blueprint and precedent, and a lack of validated clinical endpoints. The opportunities in rare disease drug development are a collaborative regulatory environment, first and foremost. Regulators understand the urgency of unmet need and the importance of collaborations with the industry to get treatments to these patient populations. And a case in point in this is some of the great regulatory guidances that have been published by the FDA in the recent few years. The study designs most commonly used in rare disease clinical trials, I would say that the most important point here is that the FDA's clear preference is a randomized, double-blind, placebo-controlled clinical trial. And there are many examples of RCTs in the rare and ultra-rare space, successful examples. Those range from simple designs like randomized parallel group design or a crossover design using the patients as their own controls, but also designs like randomized blind start design, where patients are randomized to parallel groups. And what is different about these groups is how long the patients stay on placebo before they switch in a blinded manner to active. What are the other considerations in study designs used for rare diseases? Well, when a randomized clinical trial is considered impractical and the regulators agree with you, and there is this collaborative understanding that a historical control may be a better comparator than a placebo control. There are opportunities to leverage historical controls for the rare disease drug development. These are trials where the sponsors design a single-arm clinical trial, but the comparator is a historical control cohort. By my count, in the past 5 years, there have been 6 FDA approvals that have used leveraged historical controls in one form or another in the approval process. So what are regulators' considerations when approaching such designs? The regulators expect the sponsor to show a difference between the treated single-arm trial patients and the historical controls, and that this difference is clearly attributable to the treatment itself and not to other factors. Because there is no randomization, biases can be impacting the comparisons, and these may be caused by differences in prognostic factors that may be observed or unobserved, including patients' baseline characteristics, but also external factors such as practice patterns and differences in the time and geographical location where the data were collected. To mitigate these risks, two clear steps. One is to have a very well-defined protocol for collecting historical controls, and the other is usage of propensity score-based models that essentially mimic randomization. Now let's talk a little bit about endpoints. As we discussed earlier, the rare and ultra-rare diseases tend to be very heterogeneous. This results in many times in having no single primary endpoint. And the challenge is how to capture these heterogeneity in your primary endpoint. One way is to use a composite endpoint as primary, and another way is to use multiple primary endpoints but combine them in testing. So composite endpoints. Examples of these are the so-called multi-component endpoints that are continuous scores. The components themselves may not necessarily be interpretable, but the sum of the components can be validated to be used as a scale for assessment of efficacy. A good example of that is the SARA score, the scale for assessment and rating of ataxia. Another example is the multi-domain responder index, and that's similar. However, what that has is an element of rating that is incorporated. The domains of interest for the disease are identified, and then each change from baseline from treatment to the endpoint time point is rated as -1 to connotate decline, 0 to connotate stability, and 1 to connotate improvement. And the sum of these scores make up your endpoint, the composite endpoint. Another classic example of composite endpoint is what's used in Fabry disease, and that is a combination of binary endpoints using an OR condition. Was there a significant renal effect, a cardiac effect, or a cerebrovascular effect, or did the patient die? Any of these events would trigger the binary failure criteria, and that is the endpoint that's used for establishing efficacy. When when one uses more than one primary endpoint, the ways to combine these may include the so-called global tests, such as the rank sum test and the Finkelstein-Schoenfeld test, which is a hierarchical version of the rank sum test that allows for hierarchical ranking. P-values can be combined, for example, in Fisher's combined probability test. And of course, all endpoints can be tested, and the multiplicity can be adjusted using one of the classic methods of multiplicity adjustment, such as the Hochberg method, the Holmes method, or the fallback method. And FDA has an excellent guidance on this topic. So to summarize, we talked about the endpoints, we talked about the various designs that can be used in the rare and ultra-rare space, incorporating even placebo, but sometimes using the single-arm design. And the methods used for analysis also vary. There's heavy usage, as you may imagine, of non-parametric methods, things like Wilcoxon rank sum test or the randomization or permutation test. These are exact tests that are used especially in ultra-rare. But also we use the classic methods such as the mixed model repeated measure method, which is very helpful, especially for missing data imputation purposes. And finally, there is always in rare and ultra-rare statistical analysis plans a very heavy usage of graphical techniques, patient profiles, and tools similar to that that are used to show the totality of evidence of efficacy. Here are my references. They include the key regulatory guidances that I find most helpful, as well as journal articles that I find especially relevant. Here is my contact information. Please reach out if you are interested in discussing your rare disease program, study design, execution, and analysis. Thank you for your attention.